Hi children, in this video I will be dealing with the process called photorespiration and the adaptations of C4 plants. So first let me deal with the process called photorespiration, and so that stems up with the particular enzyme called rubisco. Now talking about this enzyme, rubisco, from this very name itself you can identify its substrate and also which type of reaction it catalyzes. I have split it with the underlines. So from this you will understand the substrate is ribulose biphosphate. The substrate is what we have studied in C3 pathway. It started off with the substrate called ribulose biphosphate. So from the first part we will understand the substrate on which it acts, and the second part you can see capital C and capital O. Capital C indicate it catalyzes the reaction called carboxylation, and O indicates it catalyzes the reaction called oxygenation. Now a question arises, how can it catalyzes both carboxylation and oxygenation at one time? Yes, that is the uniqueness of this particular enzyme. This enzyme either catalyzes carboxylation or oxygenation, not both at the same time. It depends upon the relative concentration of carbon dioxide in the atmosphere. If the carbon dioxide concentration is high, definitely it catalyzes carboxylation. But if the carbon dioxide concentration in the atmosphere is low, it will catalyze oxygenation. So it is the relative concentration of carbon dioxide and oxygen in the atmosphere that decides which reaction it is going to catalyze, whether it is going to catalyze carboxylation or whether it is going to catalyze oxygenation. Now let us see what will be the outcome of catalyzing both the reactions. This side I am giving the indication that the carbon dioxide concentration is low. And this side I am indicating the carbon dioxide concentration is high. Okay. So remember the two conditions here, high carbon dioxide concentration and here low carbon dioxide concentration. Or you can talk in terms of oxygen, high oxygen concentration. Clear? If the carbon dioxide concentration is high enough, it will catalyze the reaction called carboxylation. And we know the outcome is the C3 pathway. We know the first product is 2 molecules of 3 carbon compound called 3-PGA, and finally what is the end products? We get glucose. We have analyzed the C3 pathway and we saw the end product is glucose. So if the carbon dioxide concentration in the atmosphere is pretty high, it will catalyze the reaction called carboxylation, resulting in the C3 pathway, and the first product we know is phosphoglycerate. On the other hand, if the carbon dioxide concentration is low, what will happen? It will catalyze the reaction called oxygenation. Or another name for oxygenation is photorespiration. What will happen is one molecule of phosphoglycolate and one molecule of phosphoglycerate are formed. Not only that, it ends up with the evolution of carbon dioxide, worst of all utilizing ATP. Using the ATP. So if it catalyzes oxygenation or photorespiration, we can say is a wasteful process. The reason is that two compounds are formed, one phosphoglycerate and one molecule of phosphoglycerate, glycerate and glycolate, and that will finally produce, the end product will be carbon dioxide at the expense of one molecule of ATP. Now, why did I say it is a wasteful process? It's a wasteful process because glucose is not synthesized, energy molecules like ATPA, NADPH are not synthesized, and worst of all, the ATP is utilized for releasing carbon dioxide. So, this process is not at all favored in plants. Maximum they try to avoid this particular process because the wasteful process, ATP is utilized to produce carbon dioxide, no glucose formed, no ATP, no NADPH molecule is synthesized. So, that is a very wasteful process, a bad process, that is maximum minimized by the plants by opting another pathway called the C4 pathway. Hence, C4 pathway is an adaptation to overcome photorespiration, to suppress photorespiration and to facilitate the other pathway, that is glucose synthesis via the C3 pathway. Anyway, C4 pathway produces glucose through by passing through or via the C3 pathway. So, this process is favored. Which process? The carboxylation is favored because glucose is synthesized. What is the final ultimatum of a plant is to produce glucose. So, this pathway is favored, and therefore, the C4 pathway is an adaptation. Therefore, two cells are involved, and we are going to see what and all have been incorporated as an adaptation to suppress photorespiration. So, it all stemmed up due to ribosco. Once again, ribosco is an enzyme that acts on the substrate ribulose biphosphate. You get it from the first part. And C and O stands for, it catalyzes the reaction oxygenation and carboxylation, not simultaneously, the relative concentration. The relative concentration of carbon dioxide is high than oxygen in the atmosphere. It will facilitate which reaction? This reaction. I will give it an color. It will facilitate this reaction, which is a useful process, useful to the plant because glucose is synthesized. But on the other hand, the carbon dioxide concentration goes down, it will facilitate oxygenation, which is a wasteful process. Why wasteful process? The first product is 1-phosphoglycolate and the second product is 1-phosphoglycerate, and that will finally, the end product will be formed, that is carbon dioxide utilizing ATP. So, that is a wasteful or useless process. Okay. So, it all depends on the relative concentration of carbon dioxide or oxygen in the atmosphere, and most of the plants have got adapted during the course of evolution. It has got adapted for better options. So, that is why C4 plants are very common, and not only that, C4 plants, you see almost all the plants in the tropic region are C4 plants. And because of this pathway, C4 pathway, what has happened? Photorespiration is suppressed and photosynthesis has enhanced, yield has enhanced, and very efficient the C4 plants are compared to C3 plants. So, once again going to the wordings here, photorespiration is a wasteful process as glucose, ATP, NADPH are not synthesized. So, we are justifying why photorespiration or oxygenation is a wasteful process. Why glucose or energy molecules are not synthesized and carbon dioxide is released at the expense of ATP. So, that is what is called photorespiration. Rubisco, the reason is because of rubisco. Rubisco has affinity to both carbon dioxide and oxygen. That is, in another term we can put it, it has active sites that can bind to carbon dioxide and oxygen. This binding is competitive. It is due to the presence, which is more, that reaction will be facilitated. So, it is very important. It is a competitive process. If oxygen is more, oxygenation. If carbon dioxide is more, carboxylation. The relative concentration of oxygen and carbon dioxide determines which of the two gases, carbon dioxide and oxygen, will bind to rubisco, and accordingly carboxylation or oxygenation is catalyzed by rubisco. So, it is a very competitive process. Depending on which amount of gas is high, that particular process is catalyzed. Whether it is carboxylation or oxygenation depends upon the relative concentration of carbon dioxide and oxygen in the atmosphere. So, that is why this photorespiration, there is a tendency for photorespiration to occur. It can Carboxylation or photorespiration or oxygenation can occur. If the carbon dioxide is less, it will definitely go for photorespiration, which is a wasteful process. This gives the reason why C4 plants have developed. You saw when we dealt in that previous video with C4 plants, I incorporated 2 cells. The pathway was more complicated. The reason is to suppress photorespiration. We will get into that. Adaptations of C4 plants. So, first adaptation of C4 plants is they have the 2 cell types. See, when I drew last time in the previous video, I drew the epidermis, and immediately after the epidermis I indicated the 2 cell types. One is a mesophyll cell and second is the bundle sheath cell. Why did they have 2 cell types? The reason is that light reaction occurs in mesophyll cell and dark reaction occurs in the bundle sheath cell. As a result, we know what happens in light reaction. Oxygen is evolved in light reaction. Splitting of water, photolysis of water results in the evolution of oxygen. So, oxygen will be present where? In the mesophyll cells, and dark reaction occurs in the bundle sheath cell, and we know in dark reaction our enzyme Rubisco is present. Now, Rubisco is present where? In the bundle sheath cells. Now, is there any chance of Rubisco to come in contact with the oxygen? No chance of Rubisco to come back in contact with the oxygen, because oxygen emission is taking place in a totally different cell, that is the mesophyll cell, because light reaction is occurring in the mesophyll cell and dark reaction, that is our C3 pathway, is occurring in the bundle sheath cell where our Rubisco is present. Our Rubisco can either favor carboxylation or oxygenation, but here no chance. It will not favor oxygenation, it will favor only carboxylation, because no chance of it to come in contact with oxygen which is emitted during light reaction. So, here this allows the occurrence of light reaction in the mesophyll cell. So, this 2 cell types means light reaction in the mesophyll cell and dark reaction in the bundle sheath cell. And what is the specialty of the light reaction? That should be mentioned. Light reaction, oxygen is released and dark reaction occurs in the bundle sheath cell. And what is the specialty of dark reaction? We learned that the C4 pathway is a carbon dioxide concentrating pathway. Carbon dioxide assimilation is taking place. Therefore, there is no chance of Rubisco to catalyze oxygenation. No chance, because it is not in contact with oxygen. There is no chance. Okay, the chance is ruled out, because the light reaction is occurring in a separate compartment, dark reaction where Rubisco is present is occurring in a separate compartment. So, no chance, the product of light reaction is oxygen, no chance of Rubisco to come in contact with oxygen. So, photorespiration is 0. Next, Cran's anatomy. Next adaptation in C4 plants is Cran's anatomy. So, here again I am going to draw more realistic diagram. Here we have the epidermis. Immediately after the epidermis we have the bundle sheath cells. I am going to indicate with green color, the mesophyll cells. I am just representing mesophyll cells, maybe like palisade or spongy, whatever. Okay, the mesophyll cells, and then comes the bundle sheath cells. Bundle sheath cells I am going to indicate with yellow. Look at the bundle sheet. See, one circle, I just drew only one circle, then second circle, then third circle and fourth circle. Now, look at the bundle sheet. Not only that, from this what you can infer, many layers of cells form the bundle sheet cell. Not only that, each cell is thick walled. See, so completely to say its bundle sheet cells are made up of thick walled, several layers of thick walled cells. See, several layers of thick walled, very thick walled cells. Now it gives the appearance of a wreath. What is a wreath? Wreath is that flower arrangement in the form of a circle we place on dead bodies. So it gives that appearance. So Cran's anatomy is called wreath. Wreath-like appearance. So the bundle sheath cells are arranged in the form of rings, and that too many layers of thick wall cell. Why? The advantage is that I told you oxygen is produced where? In the mesophyll cells because of light reaction. Oxygen is produced, light reaction is taking place in a mesophyll cell, oxygen is emitted. Now there is no chance of this oxygen to get into the bundle sheath cell. Why? There is no chance. It is thick walled. Because it is thick walled, it is impervious, and not only that, many layers. And so airtight it is, there is no chance of this oxygen to enter the bundle sheath cell where our rubisco is present. Here our rubisco is present. See rubisco, if I indicate with black, our rubisco is here. Inside the bundle sheath cells, our rubisco is present. If at all comes in contact with oxygen, it will catalyze the wasteful process called photorespiration. But there is no chance. Why there is no chance? Because the bundle sheath cells are thick walled and impervious to the gas. Therefore, no chance of oxygen that is produced in the mesophyll cells to get into the bundle sheath cells. So I am going to read here. The bundle sheath cells are arranged in a wreath-like manner. So to describe this wreath-like manner, it is given in bracket several layers of thick walled cells without intercellular spaces. So tightly packed. Okay. This does not allow the oxygen release in the mesophyll cells as a result of light reaction to escape into the bundle sheath cells. So there also there is no chance of the rubisco to come in contact with oxygen. So photorespiration is suppressed. Now moving on to the third point. C4 pathway is a carbon dioxide concentrating pathway. When we dealt with C4 pathway, I'll just reiterate that very step. So we saw carbon dioxide getting in, it's accepted by PEP. Then PEP immediately converts into oxaloacetic acid, the 4-carbon compound, then malic acid. Then malic acid gets into the bundle sheath cells. And then what happens is, when malic acid gets into the bundle sheath, it undergoes a process called decarboxylation, releasing of carbon dioxide and forming another product called pyruvic acid. Pyruvic acid gets across to regenerate PEP. So this pathway, see I am going to highlight it, this pathway decarboxylation, many times of decarboxylation takes place. So lots of carbon dioxide is present in the vicinity of rubisco. So a highly concentrated, it's highly concentrated with carbon dioxide. So the cycle goes many number of times, so that the concentration level is increased. Where? In the bundle sheath cell, that is in the vicinity of rubisco. So the C4 pathway is a concentrating mechanism pathway. It builds up carbon dioxide concentration. This allows carbon dioxide build up in the vicinity of rubisco. Therefore, rubisco favors only carboxylation and suppresses photorespiration. Now, can you say, is there any chance for C4 plants to undertake photorespiration? There is no chance. And when C4 plants does not undertake photorespiration, definitely a yield is high due to photosynthesis, very high yield due to photosynthesis. So they are very photosynthetically efficient plants, the C4 plants, because this is an adaptation. In 3 points I told you, the adaptation to suppress, and therefore photorespiration is negligible. So once again, a brief outline. Presence of 2 cell types, therefore light reaction occurs in one cell type, that is mesophyll cell, and dark reaction occurs in the bundle sheath cells. As a result of light reaction, oxygen is produced, but our rubisco is present in the bundle sheath cell, so there is no chance of the oxygen that is produced as a result of light reaction in the mesophyll cells to come in contact with rubisco. So there ruled out photorespiration. Second, Cran's anatomy. What is Cran's anatomy? The bundle sheath cells are arranged in the form of a wreath. To describe it, several layers of thick wall cells which are tightly packed without intercellular spaces. Therefore, there is no chance of the oxygen that is produced by the light reaction to enter into the bundle sheath cell. Again ruled out what? Photorespiration. Lastly, this pathway itself, C4 pathway itself, is a carbon dioxide concentrating pathway. The malic acid decarboxylates to form pyruvic acid and carbon dioxide. Lot of carbon dioxide, more than the atmosphere. Even a favorable atmosphere provided with carbon dioxide gas also, it's far better with the help of this pathway. It creates a carbon dioxide rich environment in the vicinity of rubisco, and therefore rubisco facilitates carboxylation at any cost. Photorespiration does not occur. This is very, very important. That's why C4 pathway is little more complicated. Anyway, the pathway through which glucose is synthesized is C3, but it takes a little more difficult path by passing through two cell types with extra products and substrates. I hope you have understood C4 pathway. Clear? And why the C4 pathway has adopted such adaptation. It is because of the process called photorespiration, which is a wasteful process. I am reiterating why wasteful. Glucose is not synthesized. Even energy products like ATP or NADPH is not synthesized. First of all, ATP is utilized to synthesize carbon dioxide. Thank you.